Hi everyone, this is Arslan Sheikh from ES19 BTech, and I will be doing a presentation on the paper Trends in Genomic Compression, written by the following paper. To start with an introduction, let the the main problem at hand is that for the genome sequencing, it's like really cheap nowadays. With the increasing technology and all those things, it has become really cheap for like even a single individual to get his genome sequenced. But the issue at the hand being that we do not have enough storage mechanisms to store such a huge amount of data, considering the silicon shortage and all the related things. That, along with how do you manage it and analyze it for scientific purposes, whatever the data is stored in the genomic sequence. So, the first thing that comes to our mind when we think of storing something is compression, and it would basically reduce the overall disk size required to store the file. And also, if the file is required to be shipped to some cloud platform, it would be easy to do that rather than just shipping the entire file size into a cloud. Take for example how for 10,000 humans, like very basic sequences of ASCII files would generate about 30 terabytes of data. This 30 terabytes of data does not include any formatting or metadata. But if we would like to include the metadata, as in the history of the genome sequence, how each base pair was sequenced, that sums up to about 20 times of the size required. That's about 600 TB. And sometimes the extra data goes on around like 30 times as well. So that would be about 1000 terabytes of data. That is like a very huge amount. So we would look into ways to tackle the problem. So the presentation is divided into four different sections, which are basically discussing the different kinds of algorithms for sequence compression. So let's start with native bit manipulation. As we know, the human genome has four major bases: A, C, adenine, cytosine, guanine, and thymine. So the thing is that we have four bases, and we are using 1 byte to store it in an ASCII format. So that is like 8 bits to store four. That's like 8 bits to store one base, which is really waste of a space. So what we can do is we could just reduce the ACGT. So we have four bases, and we could just assign 2 bits each. So in essence, we could use 8 bits to store the data of four bases. So this gives us an approximate ratio of 4:1, and but sometimes there is an error with sequencing for an indistinguishable base, for which we use some random character n or something else. So if once that is included, the encoding becomes a little more complex. To avoid that, we basically use 7 bits to encode the 3 symbols. We do not put 4 symbols into byte, we could just put 3 symbols into byte with 7 bits, and the 8th bit is left for a flag. For example, we could use the 8th bit to like denote the end of file or something like that. So the differential direct coding is a kind of encoding which basically takes A, G, C, T, and N and encodes like 3 symbols into 7 bits, as we saw previously. But with like non-standard symbols, which is not A, C, G, T, or N, it will assign 7 bits to the symbol, and the 8th bit would be basically a flag whether the byte is encoding whether the byte is encoding the 5 general bases or some additional non-standard base. Next, let's come to dictionary-based algorithms. So what a dictionary-based algorithm is like, it takes your data and keeps history of substrings with like indexes, and then we can just replace the sub whatever substrings we see with the indexes that we have seen already. The most famous examples of dictionary-based algorithms are LZ77 and LZ78. And these algorithms give us a compression rate of about 4 is to 1 to 6 is to 1. An example of these algorithms is a Splay-Tree based algorithm. A Splay-Tree is like a binary tree which like rotates its nodes to make sure the items that are recently accessed are on the top, or basically quick access. As we know, the human genome is like essential for making proteins in the human body. So the proteins have different patterns of amino acids, and amino acids come from the DNA. So the DNA is bound to have some sort of frequencies or variations, but it is also bound to have some sort of patterns, which are mostly locally prominent. So the local prominence of a sequence helps the splay trees to actually put the recently accessed elements to the top and help in more efficient encoding. Also, to detect the repeats in the sequence, a hash table is used along with the binary tree. Now let's come to statistical algorithms. Statistical algorithms are generally which take the input data and create a statistical model of it, and then present it in different forms of frequencies and other things. So basically, subsequences with a higher frequency in the genome are represented with shorter codes. So if a subsequence in a genome exists in a higher frequency, then it has a shorter code in a sense to make the effective length of the coding shorter. One of the best examples for statistical algorithms is Huffman encoding. Huffman encoding is generally used when we have large alphabet sizes and the distribution of the alphabet used is very uneven. So as you can see that this thing would not work very well for our use case, which is when we have just 4 different bases and those two are distributed evenly. So it won't really help us to use Huffman encoding because Huffman encoding in essence just boils down to bitwise encoding. So that is like a really useless for us. So we choose to avoid Huffman encoding directly on the genomic sequence. But we could use hidden Markov models for the compression. But for the compression to take place, we have to assume that it is a hidden Markov model, and we have to assume that it can be approximated by hidden Markov model. The statistical algorithms generally achieve a compression rate of 4 to 8. Expert model algorithm is like one of the statistical models, and it is basically predicts whatever the next symbol is going to be in the path, and it's very efficient. So in this algorithm, different experts are run alongside each other, and the expert with shortest possible encoding is used to encode whatever the symbol comes up next. So coming to Markov expert and the different types of experts used in the algorithm. For a Markov expert is a k-order Markov model which predicts the probability of the next symbol based on the k symbols it has already seen. The next is context Markov model. This model uses the last 512 symbols to compute the probability of whatever is going to come next. Similarly, the copy expert takes into consideration the next symbol to encode to like it takes the next symbol to be a part of the previous string whatever it has seen, and adds a certain offset to it and predicts the next value. Then there is a gene compressor model. This was proposed to encode like the non-repetitive parts of DNA sequences, and it takes the initial run where the probabilities are estimated, and then they choose the Huffman encoding. And after the Huffman encoding is chosen, it gets encoded, and then the whatever the output comes is split into blocks. And then each block is like restructured in a way that it does very good run length encoding, and this is the final step for gene compressor model. After this we have referential algorithms. Referential algorithms are kind of similar to the dictionary based approaches, except that for reference we'll just see what the difference is. So what has happened recently in the trends is that many individuals from the same species have been getting their DNA sequenced, or the scientists are doing it in one of the two ways. As all the DNA or the genome sequenced is from the same species, and we know like interspecies DNA differences are less than 1%, so they exploited the fact that the DNA of two individuals from same species is going to be vastly similar, so they designed something called as referential algorithms or referential compression. This helps in finding like long matches in the references, because 1% is like 99 symbols would be almost similar, and then one would be not similar. This kind of algorithm helps us achieve a compression rate of 1 to 400, depending on different factors. So the major factor is that it works only when you're doing sequencing within the same species. Suppose if you're trying to do sequencing on a human and a rat, it would be vastly different, because the genome would be really different because the genome of the human and the rat would be really different from each other. So to implement this algorithm, one possible way is to only store the difference between the compressed sequence, between the sequence genome and whatever we are using as a reference. The researchers have considered only three kinds of base differences: inserts, deletes, and replacements, which are like obvious by the way they have been named. So their contribution is not generally on the human genome encoding patterns, how to encode integers for absolute and relative reference positions, because to check where the base is changed or inserted, deleted, or replaced, you need an integer, and the sizes go up to gigabases and more. To check that thing, they are working on integer encoding, and they compared fixed and variable entropy coding formats, and they did report that Huffman coding is slightly better than column and alias codes. Then we come to GRS. So GRS is another referential compression tool. It's based on the Unix program diff, which gives you the longest common subsequences when you pass it two strings. So, in the given tool GRS, diff is used to compute the similarity measure between the input chromosome and a reference chromosome. If the similarity is more than a given threshold, the difference between input and the reference is compressed basically using Huffman encoding. And if the thing is does not exceed a given threshold, it is split into smaller blocks and the computation is restarted for each pair of blocks that are generated again. So this is achieved in a recursive fashion. Now coming to conclusions, the comparisons in this paper are based on the original papers, and the the issue with the original papers is that the researchers were using different set of tools and different hardwares to measure their performances, such as time or disk space or whatever they are measuring. Then the test sequences should come from different species and cover different sizes. And most of the researchers have covered just the human genome or taken a particular animal uh, which differs from researcher to researcher. And there is no specified benchmark that has some metrics to be reported. And we could, in essence, use compression rate as a factor and compression and decompression time along with the main memory usage during compression and decompression. Another usage would be in channel coding to see how we could integrate the human genome project with IoT and the cloud. So for questions I have, I think most of these are answered, but let me try it once again. Why is the genome compression different from standard text compression? So in essence, for standard text compression, we assume that it is something of with a huge alphabet size, and the genome that we have is basically limited to 4 alphabets. So it does not like really work in the similar fashion as what we would expect on a normal language or a language format. Why does a dictionary or a reference help in compression? So for a dictionary or a reference to help in compression, it needs to have a set of uh, pre-checked strings, and the since the base pairs we have are just for the dictionary and reference keep track of the previous substrings, which might sometimes be very large in size, thus drastically reducing the output string size, output sequence size. Why would standard LZ77 or not work as I said, LZ77 is basically has a sliding window, which even though given though the given sorry given that the size of the sliding window is very large, and even though we have very huge number of repetitions inside the human genome, the size of the sliding window does not make it possible to actually take into account all the repetitions, because one repetition may be left off of out of the window, and then you might have to just map it again and add it to the dictionary, which in essence increases the dictionary size by a huge amount. And Huffman coding, as I already mentioned, is a statistical coding method which does not really work well uh, for even distribution of data, and it in essence reduces back to the native bit encoding when we are looking at our topic of genome compression. That would be all from my side for today. Thank you so much for giving me your time.